Today's lecture is about the synaptic transmission that occurs between the motor nerve and the skeletal muscle. It's called the neuromuscular junction. It's a synapse, basically. It's one of the first synapses, chemical synapses, that had been studied. We know the basic organization of a synapse in the nervous system. There is a presynaptic cell, there's a postsynaptic cell, and there's a separation between them. It's called the synaptic cleft. In the presynaptic cells, the action potential causes the degranulation of neurotransmitter substance into the synaptic cleft. The neurotransmitters are normally stored in synaptic vesicles, and when the action potential in the presynaptic cells occurs, the synaptic vesicles are fused to the cell membrane, and their contents are released into the synaptic cleft. The receptors on the postsynaptic cells are activated by the receptor, so the postsynaptic cell is excited and shows action potential. This is how the chemical synapse works, basically. When we come to the neuromuscular junction, it's basically the same structure. However, there are a few important points that we must talk about. First of all, the presynaptic cell is the motor neuron. The postsynaptic cell is the skeletal muscle cell. And the synaptic vesicles contain the neurotransmitter acetylcholine. There is a single neurotransmitter in neuromuscular junction, and it's acetylcholine. And the postsynaptic receptors are also acetylcholine receptors of the nicotinic kind. They are called the nicotinic acetylcholine receptors. Acetylcholine is degraded in the synapse via the enzyme acetylcholine esterase. And these are specific details about the neuromuscular junction, which we have to consider to understand the neuromuscular junction transmission in neuromuscular junction. So the motor neuron is activated, shows action potential. This causes the synaptic vesicles containing the acetylcholine to release their content in the synaptic cleft. Acetylcholine binds and activates the nicotinic acetylcholine receptors. This in turn causes action potential in the skeletal muscle, which leads to the contraction of the skeletal muscle. This is the basic sequence of events that leads to activation of the skeletal muscle through motor neuron. The receptor on the postsynaptic side is the acetylcholine receptor on the postsynaptic side is an ionotropic receptor. Ionotropic receptors are, as you know, basic ion channels activated by a ligand. The ligand here is the neurotransmitter acetylcholine. Acetylcholine also has a metabotropic receptor, but in the neuromuscular junction we only find the ionotropic nicotinic receptor, which is called the nicotinic acetylcholine receptor. The metabotropic kind of acetylcholine receptor is called the muscarinic acetylcholine receptor, and they are both activated by acetylcholine. However, their mechanism of action is different, very different, and metabotropic receptors, as you know, are also called G protein coupled receptors. They use a G protein as an intermediary, and they increase the synthesis of intracellular signaling molecules called the second messengers, like cyclic AMP and things like that, or decrease their production. Neurotransmission through ionotropic receptors are also called the fast synaptic transmission because their activation is much faster and their end result occurs much faster than the metabotropic receptors. And also the synaptic transmission in neuromuscular junction is a fast synaptic transmission. Thus, it uses the ionotropic nicotinic acetylcholine receptors as the main receptor. When we look at the postsynaptic membrane, when the membrane of the skeletal muscle, when we and measure the membrane potential, we see that when we activate the motor neuron, this activation causes membrane potential change in the postsynaptic skeletal muscle membrane. And we see two parts of this activation. We see this bit shown with the blue arrow and also this bit. The first part is called the postsynaptic potential and the second part is called the action potential. First part, this first depolarization. First of all, we have to mention that the membrane potential of the skeletal muscle at rest is quite negative, around minus 80 millivolts. And when the motor neuron is activated or stimulated, first you see a depolarization due to the activation of the acetylcholine receptors, which is called, as I said, postsynaptic potential. This actually induces or activates the action potential. The action potential is due to the activation of basically voltage-gated sodium channels and voltage-gated potassium channels, which are located outside the synaptic cleft. The acetylcholine receptors are located within the synaptic cleft near the synaptic termination of the motor neuron. When we look at the interaction between the contraction, muscle contraction, and the action potential in the skeletal muscle, we see that the action potential is actually much faster than the contraction. The contraction follows the action potential. Action potential is necessary for the contraction to occur, and it triggers the contraction. But when the action potential occurs, there is a delay between the start of the muscle contraction and the start of the action potential. And action potential also is much shorter than the contraction, duration of the contraction. For action potential is about 10 milliseconds in skeletal muscle. However, the contraction takes between 20-30 milliseconds in the skeletal muscle. So the contraction is transient, and actually it's a very fast contraction and relaxation event when you look at it. It's 20-30 milliseconds are very short periods of time, and it's called a twitch. So skeletal muscle is basically contracts by twitching. This is also the case for the cardiac muscle, because cardiac muscle and the skeletal muscle, they are both striated muscles. However, smooth muscles can actually produce sustained contractile force, which lasts minutes or even hours. However, for the striated muscles, they cannot produce sustained contraction force. They can only produce transient contractions, which are called twitches. Thus, how does the striated muscle, skeletal muscle, produce sustained contractions? There are two mechanisms for that. The first is called the summation, and the summation is basically twitches that come one after the other. If you stimulate the cell at a high frequency, if you produce action potentials one after the other, before there is enough time for the muscle to relax, the contractile force of these consecutive stimulations actually start to add up, which is called addition, which causes some sort of a continuous force production or continuous contraction. The second actually way, or the second mechanism for inducing or for producing sustained contractions in skeletal muscle is asynchronous contraction of the so-called motor units. So the organization of the, as you may probably know, that the organization of the skeletal muscle is each motor nerve fiber ends up or terminates in a single skeletal muscle cell. So each skeletal muscle fiber cell is stimulated by a single nerve fiber. There are millions of such units in an anatomic muscle, skeletal muscle. So these units do not contract all together. They contract asynchronously. So when one of them contracts and relaxes, a second one, a different one, contracts. So it ends up with a continuous force production, contractile force production in the skeletal muscle. This is also the second way to produce sustained contraction in the skeletal muscle. But the important thing is, as the skeletal muscle can only produce twitches, which means that it has to reset itself at the end of each twitch. So the membrane must be in a state that can be activated at the end of each twitch. This requires the reactivation or changing of the voltage-gated sodium channels from inactivated state to an activatable closed state, which requires the membrane potential to return to negative resting levels for a period of time. Otherwise, the cell won't respond to further depolarization and cannot produce action potentials. Which means that the acetylcholine stimulus must be transient also. So the release of acetylcholine is quite short-lived. The acetylcholine releases into the cleft as a packet, and then immediately the acetylcholine is degraded by the enzyme acetylcholine esterase. So the period of acetylcholine staying in the synaptic cleft is very short. As you can see in the lower graph, this period is denoted by the horizontal blue bars. That's the period where the acetylcholine stays in the synaptic cleft. If you prevent the skeletal muscle to produce an action potential, you end up with the postsynaptic, only with the postsynaptic potential. So the lower graph shows such a cell. The upper graph shows a fully functional motor neuron skeletal muscle unit. So when you stimulate the motor neuron, you release acetylcholine into the synaptic cleft, and you see in the lower graph a transient depolarization, which in the upper graph ends with the action potential and also the contraction. As you can see, between the consecutive stimulations, the membrane potential returns back to its negative resting value until the second package of acetylcholine is released to the nerve action potential. However, if you block the nicotinic acetylcholine receptor, for example, using curare as a chemical that blocks the acetylcholine receptor and nicotinic acetylcholine receptor and prevents the action of acetylcholine on this receptor, which basically what it does is it prevents the postsynaptic depolarization and prevents the production of action potential and contraction. This has some usage in medicine, which for the surgical operations, you need not only general anesthesia, but you also need muscle relaxation, skeletal muscle relaxation. You can obtain this by either blocking the acetylcholine receptor, nicotinic acetylcholine receptor, with curare-like molecules, which is called a hyperpolarization block, because the membrane stays hyperpolarized. Even if you stimulate the nerve, the motor nerve, and the motor nerve functions and releases acetylcholine, this does not produce a depolarization action potential in the skeletal muscle, postsynaptic skeletal muscle. The postsynaptic skeletal muscle stays hyperpolarized. This state is called hyperpolarization block. So there's another way, which is a little more complicated to understand. You can use some drugs or substances which blocks the acetylcholine esterase enzyme. So the enzyme cannot function and cannot break down the acetylcholine. So the acetylcholine concentration within the synaptic cleft increases as acetylcholine accumulates due to consecutive release of acetylcholine by nerve activation. In this case, what you see is rather interesting, because in the fully functional motor unit, you see at first there is an autonomous action potential production, which is not directly related to the acetylcholine release first, and then as time goes by, the membrane starts to depolarize and do not respond to acetylcholine release or the nerve stimulation anymore, and the action potential stops, the contraction stops. However, the membrane becomes depolarized. This is called a depolarization block, and which is induced by acetylcholine esterase inhibitors, like the drug or the compound neostigmine or physostigmine. This group of compounds causes this depolarization block. The reason, to understand the reason behind the depolarization block, we have to go to Goldman-Hodgkin-Katz equation. You remember Goldman-Hodgkin-Katz equation, I believe. And when we write the situation for the neuromuscular junction, we have the potassium conductance, we have the conductance due to acetylcholine receptor activation, and we also have the conductance due to voltage-gated sodium channels. So Na is the voltage-gated sodium channels, ACH is the conductance due to nicotinic acetylcholine receptors, and GK is the conductance due to potassium channels that are active during the basal resting state conditions. And as these voltage-gated sodium channels pass sodium ions, the GVNA is multiplied by the sodium nernst potential. The GK is multiplied by the potassium nernst potential, equilibrium potential. However, the acetylcholine, nicotinic acetylcholine channels are not potassium or sodium selective channels. They are non-selective cation channels, so they are multiplied by the reversal potential of the cations. If you take the cations as a whole, the reversal potential for the whole cations is around, as the concentration of the cations inside and outside the cell is almost equal, you end up with a nernst potential around 0 or minus 10 millivolts. So this is the setup of the Goldman-Katz equation for the synaptic transmission. In the resting cell, the main the conductance that is dominant is the potassium conductance. So the membrane potential is around minus 80 millivolts, which is very close to potassium nernst potential, the equilibrium potential. When the synapse is activated and the acetylcholine is released into the synaptic cleft, the acetylcholine receptors gets activated and they open up. So acetylcholine receptor conductance becomes important. So the membrane depolarizes towards minus 10 millivolts. How much it depolarizes is dependent on the ratio between the GK and GACH. More activation on the acetylcholine receptor side, you get more depolarization. So this is the first depolarization, the postsynaptic depolarization, which leads to action potential due to activation of voltage-activated sodium channels. This depolarization activates them and they open up, and their conductance are much greater than the rest, the GK and the GACH, so they dominate the membrane. The GVNA dominates the membrane, and membrane potential goes to very close to sodium nernst potential, which is around plus 50 millivolts. Eventually, this is the action potential. Eventually, the sodium channels get inactivated, as you know, and also voltage-gated potassium channels open, which we do not consider or discuss here to keep things simple. However, at the end, the sodium and voltage-gated sodium and voltage-gated potassium conductances goes to 0 at the end of the action potential, and things return to starting value. If you use a blocker of the acetylcholine esterase enzyme, then the acetylcholine concentration in the cleft will increase, and as the acetylcholine accumulates in the cleft, nicotinic acetylcholine receptor conductance do not goes back to 0, even if the nerve activation does not occur. So, which means that the membrane cannot return back to minus 80 millivolts, but stays a little bit depolarized, because there is also another conductance at this situation, which depolarizes the membrane, as you can see here. The thing we should notice here that even though the acetylcholine increases with time, membrane potential in the lower graph does not increase indefinitely. It increases to a certain level and then it saturates, even if the acetylcholine concentration is increasing in the cleft. We can understand why this is happening, because here you can see that the extreme saturation where you can get under these circumstances is that the dominant conductance will be the acetylcholine receptor conductance. Eventually, if the acetylcholine concentration increases to a very high level and all the nicotinic acetylcholine receptors are activated, then you'll end up with a very large acetylcholine receptor conductance, which will dominate the membrane, which means that it will only depolarize the cell to the cationic reversal potential, which is minus 10 millivolts or 0 millivolts, which won't go further than that. So this is the maximum depolarization you can get under these conditions. The thing is, if you depolarize the cell and do not allow it to repolarize to minus 80, 90 millivolts back to resting levels, then the sodium channels, the voltage-gated sodium channels, which open and then inactivate, as you can see here, cannot go back to closed state, so stays inactivated. For them to go back to closed state, as shown with the red arrow, you need to go back to the resting negative membrane potentials. You prevent the cell under these conditions. You prevent the cell from going back to negative potentials. Thus, you prevent the voltage-gated sodium channels to go from inactive state to closed state, which prevents them from responding to the voltage changes anymore. So even if you can continue to depolarize the cell, as you can see in this graph, even if you continue to depolarize the cell, after a certain level of depolarization, the voltage-gated sodium channels do not respond to this depolarization and cannot produce an action potential, because most of them are in the inactive state and they cannot go back to closed state and reopen. Thus, this is basically the mechanism how the depolarization block prevents action potential. There is also another mechanism which is a little bit more difficult to understand. You can see here the reason for that. If you think about the effect of voltage-gated channels, it's the GVNA increases, and then it means that the membrane will depolarize towards E sodium, which is plus 50 millivolts. But however, for this to occur, the GVNA should be comparable to the other conductances. GVNA is quite large, so in normal conditions, compared to both to GACH and GK. However, if you accumulate acetylcholine in the synaptic cleft, the GACH will increase quite a bit and becomes comparable to GVNA. And this means that GVNA cannot depolarize the cell as much as it used to. So the membrane cannot be depolarized to plus 50 millivolt to E sodium anymore. This is also another mechanism how the acetylcholine esterase inhibition or depolarization block prevents the action potential production and muscle contraction. This is utilized by the surgeons or the anesthesiologists to prevent the muscles from contracting during surgical operations. They administer this acetylcholine esterase drugs, inhibitor drugs, into the bloodstream of the patient, and during the operation the muscles relaxes, even though the motor neurons are still active and functioning. The muscles do not contract, and at the end of the operation they stop the administration, and the drugs is eliminated from the bloodstream, and the acetylcholinesterase regains function, because these drugs are reversible inhibitors, and then the neuromuscular junction starts to function as normal. And sometimes, to hasten this reversal, you can use curare-like drugs to block the action of acetylcholine in the receptor, which reverses the depolarization block faster. If the inhibition of acetylcholinesterase enzymes are irreversible, some other drugs also can induce irreversible inhibition. This causes constant paralysis of skeletal muscles, and also the muscles used in breathing. And these are toxic substances, which are called the nerve agents and used in the warfare, or also as insecticides against the insects. That's why the insecticides are very poisonous for mammals and human beings. Okay, this is the basics of neuromuscular transmission. See you in the next lecture.